DaeSung Microbiological Labs always puts quality first, building trust with every customer. As a specialized manufacturer of veterinary vaccines, we are committed to excellence at every step of production. With independent production systems for both killed and live vaccines, we maintain rigorous quality control at every stage. Also, separate access points are established for swine and poultry antigen production areas to ensure separation of personnel flow. The viral vaccine team's production area is also operated to maintain separation between cell culture and virus culture processes. Each team maintains independent facilities and separate utility systems, ensuring the production of safe products without cross-contamination. DaeSung Microbiological Labs produces vaccines in 3 stages: clean manufacturing environment construction process, cultivation, and finished production process. Both killed and live vaccines go through the process of clean room preparation stage. Cultivation is carried out by separate teams, and each team handles cultivation according to the type of biological material. Finished production process also differs, with distinct procedures tailored to each vaccine type. DaeSung Microbiological Labs operates a fully equipped clean manufacturing environment designed exclusively for vaccine production. For the production of veterinary pharmaceuticals, we generate both water for injection and purified water, which are supplied through an integrated water distribution system to each manufacturing unit. All production equipment is sterilized using either built-in sterilization systems or autoclaves. Independent facilities for killed and live vaccines operate under advanced HVAC systems. Air cleanliness, temperature, humidity, and pressure differentials are maintained through an automated building management system and continuous environmental monitoring. To prevent cross-contamination, the cultivation process takes place in strictly controlled facilities, divided into 3 independent teams: bacterial vaccine team, viral vaccine team, and poultry vaccine team. The bacterial vaccine team cultures bacterial strains in optimized media. Master and working seeds for vaccine production are stored separately in seed storage freezers and handled only by authorized personnel. Manufacturing operators retrieve bacterial strains from the freezer and streak them onto solid agar media inside a biosafety cabinet. The inoculated plates are incubated in a temperature-controlled room, and then bacterial colonies are transferred into suitable liquid media for further culture. The bacteria, cultured and amplified in incubators, are transferred to fermenters for further cultivation under optimized conditions. After cultivation, the bacterial culture undergoes centrifugation, disruption, purification, and concentration to produce the final bacterial antigen for vaccine production. Unlike bacteria, viruses require host cells for propagation. To manufacture cell culture-based vaccines, frozen cell lines stored in liquid nitrogen tanks at -196 degrees Celsius are first retrieved and then thawed for culture. Cells are progressively expanded through either adherent or suspension culture until they reach quantities required for vaccine production. During cell culture, the condition and number of cells are closely monitored. When subculture is completed, production seed virus is inoculated into cells. After inoculating virus, cultivate it in an incubator. Once cultivation is completed, the virus culture is harvested, purified, and used in vaccine production. For poultry vaccines, a dedicated poultry vaccine team carries out the cultivation process separately. First, fertilized eggs that have developed to specific embryonic age are candled to identify and remove non-viable embryos. After disinfection, viable eggs are inoculated with virus using semi-automatic injectors. The inoculated eggs are placed in incubators, where temperature and humidity are set to the ideal conditions to allow the virus to propagate fully. After incubation, the eggs go through second candling to discard dead or contaminated embryos, and the remaining eggs are cooled in a cold chamber. After 12 hours of cooling at cooling room, the virus culture is harvested using a semi-automatic harvester. The harvested virus culture is then filtered and concentrated to produce the final virus antigen for vaccine production. The filling process for killed vaccines and live vaccines are carried out in separate facilities, with each utilizing its own dedicated system. The process for killed vaccines begins by inactivating bacterial or viral cultures with the appropriate inactivating agents to prepare the antigens. The inactivated culture is then mixed with adjuvants in manufacturing tanks. Before filling, polypropylene bottles, commonly used as containers for killed vaccines, are sterilized using EO gas. The bulk product is transferred to filling tank, along with filling line and automatically filled into containers. Once filling is completed, the containers are stoppered with rubber closures. The containers stoppered with rubber closures are sealed with aluminum caps before labeling. Live vaccines, typically produced as lyophilized formulations, require the proper preparation of a lyoprotectant. The lyoprotectant is mixed with the virus culture in manufacturing tanks. Glass vials, the primary containers for live vaccines, are washed in vial washers and sterilized in tunnel sterilizers at 300 degrees Celsius. The bulk product is automatically transferred to the filling tank and filled into the container. These containers are sealed halfway with rubber closures and transferred to the lyophilizers for freeze drying. Within aseptic lyophilization chambers, an automatic loading and unloading system operates, and once the freeze drying process is complete, the containers are fully stoppered with rubber closures and automatically discharged. The containers stoppered with rubber closures are capped with aluminum caps and labeled as finished products. All labeled finished products are stored in refrigerated warehouses and go through national quality testing and approval before final release. DaeSung Microbiological Labs complies with global standards and regulations, ensuring that only the highest quality products are delivered through advanced facilities and a rigorous quality assurance system.